The topic of this webinar is Personalizing Medicine, an introduction to pharmacogenetics. I'm pleased to introduce the speaker of today, Dr. Alison Ferber, who brings a wealth of experience and insights to today's discussion. She holds a PhD in genetics and has built her career at the intersection of science and industry, with experience in academia and at 2 leading genetic testing companies. Alison now serves as the marketing lead at a census, where she leverages her scientific background to shape communication and strategy in the rapidly evolving field of genomics.
Thank you all for joining us today while we talk about pharmacogenomics. And I'll start, of course, with some quick objectives so you can know where we're headed in this next 30 minutes. We're going to start with just understanding what I mean by pharmacogenomic testing. Then we're going to identify some pharmacogenomic resources that will be useful to all of us. And the most important part is we're going to talk about the clinical value of pharmacogenomic tests. All right, let's start with the basics. What do I mean when I say pharmacogenomics? This is really simply just the study of how an individual's genetic makeup influences their response to medication. Now, you may have heard the term pharmacogenomics or pharmacogenetics. What's the difference there? We can think of pharmacogenomics as how all the genes in the genome influence drug response. And then a subset of that would be pharmacogenetics, where we're simply talking about how one gene influences the drug response. So for the purposes of our talk today, we can kind of use them interchangeably, just a difference of scale. But for simplicity's sake and to save on syllables, from here on out, I'm going to refer to both as PGX. So make it a little easier for all of us. Okay, now that we know what it is, the important question, what do we do with it? So if PGX is everything in this green box on the left, how the DNA influences drug response, and that could be through changes in absorption, distribution, metabolism, or excretion of drugs. If that is PGX, then how do we get from there to changing our actions in the clinic? How do we use this information to impact our drug selection choices or our dosing choices and our monitoring choices? Because in the end, the only goal of having this test result is to improve patient care. And by that, I mean with measurable things like increasing efficacies of our medications and reducing adverse events. So I think it goes without saying that this is the goal anytime anyone writes a prescription, improving patient care. But we need to take a small step back and think, what is going to be the impact of specifically pharmacogenetic testing on the general population? What should our expectation be? So I've created not really a graph, more of a picture of 8 studies from 8 different countries who took a general adult population and did pharmacogenomic testing on these people. And they found that statistically, we can say everyone carries at least one actionable PGX variant. So, maybe not literally everyone, but more than 99% of the participants in most of these studies carry an actionable variant. So we see some variability. This will depend on the genes that were tested in each study, in each PGX panel. And we do see variability in the types of variants that are detected across countries or different ancestry groups, which is interesting. And we'll come back to briefly later. Also, I want to mention, I do have references for these at the end if you want to download that handout that Philip mentioned. Okay, so essentially, we can just say that everyone carries a PGX variant, but what are the likelihood that each of us will be prescribed a drug in our lifetime that will match up with that variant? That's kind of the other side of the coin in this situation. So I've referenced here 10 years old. It's a little bit old in the field of genetics, but this is a study that's regularly referenced, and you'll see again and again, and we can use as a baseline. So this group found that 7% of FDA-approved medications, we're thinking about the US here, have actionable PGX variants. So I would use this as a minimum. Studies have shown now that in this last 10 years, year over year, the PGX variants, the newly approved FDA drugs each year, a higher proportion of those year over year will have actionable PGX variants. So in the last 10 years, this proportion of FDA approved medications with PGX variants has probably grown. And so it's a higher slice of the pie. Now, some of these drugs are going to be to treat very specific symptoms or very specific conditions that might be rare. But some of these drugs are for very commonly prescribed medications, and we'll come to that. So if we think of that, maybe 10% of drugs have actionable PGX variants, this will translate then, because some are so common, into 18% of outpatient prescriptions will be affected by actionable PGX variants. And again, this number will fluctuate depending on the type of cohort you're looking at. This is outpatient, so that's probably a little lower than if we looked at an inpatient or oncology cohort. So maybe for us, we'll just say 1 in 5 moving forward. Okay, so a fair amount of prescriptions are going to be impacted here by these test results. So the next question then becomes, how do we select which medications that we are able to say have actionable PGX variants? And this is done by looking for consistent results from well-designed, well-conducted studies that demonstrate a clear link between a gene variant and a drug's effect. So that sounds very logical and a really good place to start. But I think you can easily imagine that in practice, it becomes complicated to find all that primary research and have the experience and knowledge to determine if it's well-designed and well-conducted study to begin with, and then if the study has established that clear link. So lucky for all of us, there are many groups who have been working on this and already come to conclusions. So I have a list of resources here. Now, first, many countries across the world have national guidelines for their physicians for which drugs and genes should be included in actionable PGX tests. So you can Google that for wherever you're interested in. But more generally, the FDA puts out a table of pharmacogenomic associations for drugs where they believe have therapeutic recommendations. The FDA also provides box labels for drugs where they either require or recommend PGX testing. So that is a great place to start. We also have the Dutch Pharmacogenetic Working Group. This is a multidisciplinary organization that works to collect, curate, and then make publicly available pharmacogenomic data with the goal of bringing it accessibly to the clinic. PharmGKB is another similar resource, freely available, funded by the NIH, that brings together PGX data and makes it available. But what we consider the gold standard of PGX guidelines is the Clinical Pharmacogenetics Implementation Consortium. So for that reason, I'm going to take a minute and talk about CPIC a little more specifically. So CPIC is an international consortium that develops and publishes freely the evidence-based peer-reviewed guidelines that are designed to help clinicians understand how genetic tests, number one, should be used and how to optimize their drug therapy. So this ends up producing two things. Number one, assigning evidence levels, linking that genotype to phenotype. So based on the primary literature, how sure can we be that there are consistent results linking genotype to phenotype, back to that selection definition. And number two, and probably most importantly, CPIC provides detailed gene drug clinical practice guidelines. So let's start with how they assign evidence levels. So CPIC has a 4-tiered system that classifies these gene drug pairs based on, number one, the strength of the evidence, but also the actionability of the recommendations. So this looks like classes A and B and C and D. So A and B, these are gene drug pairs that have a clear link between the genetic variant and the drug effect. They have some available prescribing action, and the alternatives are highly likely to be safe and effective. Now, these are the genes and drugs that you are most likely to see on a PGX report. These are the ones we want to include that have the high levels of evidence. Now, levels C and D, these are where the evidence is still insufficient or maybe inconclusive, or it could be a pair that has no actionable recommendation or no clear alternative. So these are genes and drug interactions that would not be included or should not be included on most PGX reports because they're not actionable and we can't be sure about them. Okay, I wanted to just show you briefly how this looks. If you go to the CPIC website, and I'll give you a link at the end, it's a very accessible table, which I think is really important. PGX test results should be accessible to as many patients and physicians as possible. The table is simply the association between the gene and the drug, a link to their guidelines, and then their evidence level and the status of that analysis. And then they bring in that external information that we talked about from PharmGKB, has their own evidence level, so you can find any contradictions. They bring in that FDA box label, so we can see really clearly where testing would be required versus recommended, or just simply actionable. And then they have links to all of that primary data that they use to come to that decision. So this is a really fantastic resource. Okay, so what they have here, if we think about just evidence levels A and B, obviously in the table you can see all of them, are currently 105 genes that span the medical specialties from cardiology, oncology, neurology, and even everyday general practice would prescribe some of these. So to give you an idea of some of the more common everyday drugs that would be included in this list, I have organized a few by what they would be used to treat, and we'll come to it later. Medications used to treat depression lend themselves very well to PGX because it eliminates the trial and error as well as side effects. There are pain medications that are included as actionable PGX drugs, including, and also then statins, which would be used to lower cholesterol, are PGX actionable, and proton pump inhibitors that would be used to treat acid reflux are quite actionable with PGX. Okay, so we've hit the middle point, and I want to summarize, even though we haven't been together long. But what we have established here is that just about everyone will carry an actionable variant, and at least I would say 1 in 5 prescriptions will have a PGX recommendation. So we can see that the need is broad and that the potential for impact is great. So the question then becomes for the second half, how do we bring this PGX testing into practice? Okay, I want to start by thinking about how PGX test results might make their way to you, or what types of tests are available currently in the market. So first, you might come across a person who has ordered their own direct-to-consumer test. So this would be where a patient sends in their own sample to a company and gets some test results. Now, this is a fantastic option because it increases the accessibility. The more people that have this information, gives us more potential for making those prescribing decisions. Now, the downsides can be that these direct-to-consumer testings companies might not have the accreditations of a diagnostic lab. It could be variable. And so the regulation, we can't be sure. We would have to find out. And that variability in regulation can lead to a variability in accuracy. That's the true problem. In addition, these companies can include any genes on their panel and have test results, maybe not in evidence levels A and B. Maybe they dip down into C and D. Maybe they leave some out that you would have preferred to see. So what is in the report and how the report is displayed to you is going to be different across every company, which is going to make the clinical integration more difficult when it comes time to write that prescription. So the second type of test that I thought of would be when the PGX results are secondary to a diagnostic test. Now, the positives here would be that a diagnostic test would come from a lab that is accredited, so the accuracy of these results will be higher. Because these are results that are secondary to a larger test, there'll be a larger body of data, so reanalysis of this information will be possible down the road when new gene-drug associations become available. The problem here is this may not be a test that you ordered. So therefore, integrating it into your clinical practice and workflow might be difficult, especially as the test results and how they're presented can be different across companies. This might be a challenge. So the third way that you might get PGX test results would be if you yourself were ordering a prescription-specific test. So this would be a situation that before writing the prescription, you would like to have that PGX result. So again, this is going to be a diagnostic result from an accredited lab. So the accuracy will be high. This is a special situation because in the other two, those patients might not be on any medications and so there's no immediate action to be taken based on the result. But because this test was ordered specifically for a prescription, it will be highly relevant immediately to your patient. It will be useful. And again, because you ordered it for that reason, it will be integrated easily. Now, the downside, I would say here, would be the wait time. When you decide to write a prescription and now you need to order a test at PGX, there will be some amount of waiting, and that can be inconvenient, whereas the other two already exist with the patient. And here, if we order, let's say, a specific panel on statins and get the PGX information on that, maybe a few years later, that patient now would like a prescription for an antidepressant, and we can't go back to that statin data and reanalyze. We would need to redo the test. So that is one of the limitations of prescription-specific testing. Okay, but whichever one of these three ways the data comes to you, the structure and the main components of the data will be the same. We will have information, obviously, on which medication we're talking about, and then the genotype. So that's the change in the DNA specific to the patient. We'll have a phenotype. These are the observable characteristics resulting from that genotype. Usually there'll be some interpretation. So this is a summary of the genotype-phenotype association, including possible risks. And then the most important part is the recommendation. So these are the specific drug selection or dosing guidelines that are going to be based on that individual's genotype. So let's start by what do I mean by genotype? How is that going to look on the report? Okay, a genotype on a PGX report can be displayed one of two ways. It can be directly genetic variants. So that's a change in the DNA sequence of a gene, as we just said. And it could look like one of these two examples. So here we have a change in the gene RYR1, and we give the cDNA position and the nucleotide change. So that defines the specific place we have a change. Or sometimes we can display this as an RSID, which is just a unique identifier that defines some specific genetic variant. Now, those are the variants. Those are going to be causing changes in gene function. But if we think about how we would describe normal function genotypes, then we're going to use words like reference or wild type when we talk genetic variants. But in PGX, we have a special system called star alleles that we use to identify genetic variants. So the first reason we do that is it simplifies things a little bit. Physicians don't need to understand cDNA or protein changes or this RSID. We can just see the gene and the star number. So in this case, the normal function allele will be star 1. So we have an example here of a normal function CYP2C19, which is star 1. And then any other subsequent number would lead to a difference in gene function. So here in CYP2D6, we have star 2 and star 17. The other benefit of star alleles is that these star numbers might not correlate to just one genetic variant. CYP2D6 star 17 actually indicates 3 specific genetic variants all at once. So they're, again, simplifying that report for people to really understand what's going on. Okay, so that's the genotype, obviously the starting point, but then we need to understand the phenotype that's associated with that genotype to move forward. So if we imagine we have a genetic variant in a gene, and that gene is going to create a protein, we want to know what that variant is, how that variant is impacting the function of that protein. So really basically, we can change the functional status. We can increase, have normal function, decrease, or no function. But making it just a little step more complicated, because many of the PGX genes actually make proteins that are enzymes. What we like to say is what is the impact on the ability of that enzyme to metabolize the drug. So one of those subsets of gene function, gene function that I mentioned earlier. So when we have a gene that creates an enzyme, we use a little bit different language: ultra rapid, rapid, normal, intermediate, or poor metabolizer. But essentially, it's meaning the same thing in function. All right, I want to give an example of how this looks in practice. So this is the CPIC recommendations for the gene CYP2C19. And this is the impact of that gene on proton pump inhibitors. So we get very specific. So because CYP2C19 is an enzyme, we're going to use that language of ultra rapid to poor metabolizer. I have, in the paper that they provide, they will give recommendations for all 5 different functional groups, but here I've narrowed it down just to the 2 extremes, the ultra rapid and the poor metabolizer. So if we look first at the ultra rapid, let's understand the implication of that phenotype, right? So we've come to the label of ultra rapid metabolizer based on this individual's genotype. And now the implication of that is that this person will have a decreased plasma concentration of PPIs compared with a normal metabolizer. And the risk of that is that they might have an increased risk of therapeutic failure, right? They have a lower level of circulating medication than we would expect. So the recommendation then in that case would be to start with an increased dose, even dividing that dose across the day and then monitoring. So the other end of that spectrum is a person whose genotype would indicate that they're a poor metabolizer. So this person will have an increased plasma concentration of PPIs compared to a normal metabolizer. So this will lead to a risk of increased chance of efficacy, which could result in potential toxicity. So the recommendation here would be to start with the normal dose, the standard dose, and then for chronic therapy, decrease that dose over time and monitor here for efficacy. So the recommendations are very specific. I've referenced the paper here, but for each of these gene drug combinations, CPIC publishes a paper with all of the information that you would need, which is very nice. Okay, so we have talked about what exactly we mean by pharmacogenetics and how that can influence prescription medic selection and dosing and how we monitor. But the real goal is to use this information to improve patient care. So we can do that. We can measure that in one of 3 ways. So first is increased efficacy. Now, how we measure efficacy of a drug will be different depending on the indication for that drug, what specialty we're in. But there have been papers, in many different specialties, illustrating the efficacy of PGX test results. Now, I picked one for patients who were diagnosed with depression, and they were measuring response rate. And they found a significantly increased response rate in the patients who had prescriptions based on the PGX test results. So the second, and very importantly, is a reduction in adverse reactions. So this was a general study that I've selected here across many sites in Europe. And these patients were not selected based on their diagnosis. It was just adults. And even in that population, they found a significant and 24% reduction in clinically relevant adverse drug reactions. So taken together, both of these things can lead us to our third and secondary benefit, which is saving time and money. We save time by not going through the trial and error of finding a drug that works for a specific person. And now we save resources and avoid these adverse reactions. So this last paper was a meta-analysis of 108 studies that found that 71% of them found that PGX testing was cost-effective, cost-saving in their hospital or clinic. So I think that we can see from this, the potential for PGX is very large, and we need to find a way to utilize this test for the patients that need it when they need it. But I want to acknowledge the limitations really short here before we leave you. So I've listed 4 of the limitations I've kind of touched on in the whole talk. So first, there where we started out, there are a limited number of drugs that are included currently as PGX actionable. So as I already mentioned, these new gene-drug associations are constantly being established. As new drugs enter the market, they are increasingly more likely to be impacted by PGX results, but also as new studies become available, that information is integrated in the current knowledge, and existing drugs can have their evidence levels increased to A or B and now be actionable in the clinic. Okay, a second limitation that I feel like we're addressing here is a lack of education to interpret and apply PGX results. So this is a very new test, relatively speaking, and these are results that most physicians have never seen before. So luckily, all over the world, all sorts of hospitals and health centers are developing targeted educational resources. You go on the internet, there are fantastic videos, handouts, entire papers for both physicians and patients to understand what PGX testing is and how to use it. We're really in a good spot today to educate ourselves. Okay, another limitation that I touched on was the underrepresentation of diverse populations. So like most genetic research, European cohorts are used first, but this bias in ancestry could lead us to not making the correct recommendations in those other populations. But the field has acknowledged this limitation, and there are already initiatives underway to broaden representation and increase diversity of our databases, making those recommendations much more accurate. So finally, and as I already mentioned, it can be difficult to integrate these test results into daily care and clinical workflow. Do you have the test result available at the moment you write the prescription? That's going to be our limiting factor, I think. So to this end, the field generally is working on the development of electronic health record integration in countries that have electronic health records. You can read very good papers from the NHS about this. Or more generally, there's a lot of work being done for PGX passports. This would be information that patients carry themselves and integrate in their own physician to have that information available when they're ready to write a prescription. All right. So I want to summarize the 4 take-home points of what we talked about. Definition of PGX, how an individual's genetic makeup influences their response to drugs. Pretty simple. We have talked about how actionable PGX variants are found in nearly all people and impact at least 1 in 5 prescriptions. We've talked about some resources like CPIC that summarize the existing evidence and provide recommendations. And I hope I've convinced you that changes in medication or dosage based on PGX results can improve patient outcomes. With that, I'm going to leave you some resources that we talked about at the top, and then 3 more just fantastic educational resources. You can download this in the handout if you have additional interest. And with that, I want to thank you for your time, and I'm happy to take your questions.
Great. Thank you very much, Alison. I'm going to now check the comments and questions. Anybody feel free to answer or to make a question? Here we got one. Somebody is asking the following. You said that PGX results could be secondary to a diagnostic result. what kind of test would that be?
Yeah, that's a good question. So I am speaking of a genetic test result that would be large. Like a whole exome or a whole genome test result would detect variants in genes beyond that diagnostic question. And some companies do include pharmacogenomic test results as a secondary finding. So like at our census with My Life Genome, our whole genome sequencing test, because we sequence the whole genome, we are able to then additionally detect pharmacogenomic variants and include them in the report. And there are other companies that do something similar. So yeah, we're thinking about large diagnostic tests like WES or whole genome sequencing in that situation. Thank you.
Thank you. So let's see if anybody else has some ideas or questions. Somebody is asking, are PGX tests only relevant for specific specialties?
Yeah, okay, so like specific medical specialties.
Mm-hmm.
Yeah, that's a good question. So I think when we think about required testing, then maybe that would fall more in an inpatient setting or for specific diseases like cystic fibrosis in oncology. But really, given in the examples that I've shown, it is true that all specialties could expect to write prescriptions. Even a general or family practice doctor could be writing a prescription for a medication that is actionable with PGX. So I do think that all physicians should at least have some awareness that their prescriptions could be impacted by PGX. Yeah, that's a very good question.
Yes, that's pretty relevant. Okay. I'll see if anybody else wants to comment. There is another question coming from the audience. How do I explain PGX results to my patients in understandable terms?
Yeah, yes. I think I want to start by saying that the goal of PGX should be that the results are so understandable that physicians and patients don't need a specialized genetics understanding to act on them. So we're not there yet, I think, as a field. But what I like to think about PGX when I would explain it to a person with no medical knowledge is start from what they know. We all know that people respond to medication differently. Like everyone is familiar with that concept. So some of that might be your environment and lifestyle choices, but now we can tell patients, we know that at least some of that reaction will be because of your genetic makeup. And while we all have the same genes, each of us can have different variations of those genes, different flavors of genes. And this test will find that. But if you are interested in that, there are fantastic videos on the internet, actually, role playing that conversation between a patient and a physician, because this is a question that everyone is facing right now. And there are really good resources. Yeah.
Yeah, totally. Okay. Thank you for that answer. I'll check if anybody has any more questions. All right. Let me see this one. Are these results static over a lifetime or can they change? This is also an interesting question.
Yeah. Okay, yep, that's fantastic. So all of these test results are from what we'd say the germline genome. So these are variants that a person was born with and will have their entire life. So their variants won't change. And what we've established with high evidence as those genetic variants impacting specific drug response is unlikely to change. But what we understand about that interaction between the genome and drugs will change over time. So we will have more drugs or maybe refine our understanding. So while fundamentally these do not change over a lifetime, as more information becomes available, how we interpret the results will change over a person's lifetime. I hope that was clear.
Yes, great. Okay. Well, I think we got no more questions right now. I think we should close the session now.
Thank you. Yeah, I was going to say, if we've missed your question from the questions, I will follow up by email. I'm sure
Yes,
everyone
as said before, yeah.
As mentioned, yeah. Thank you.
I'd like to thank you very much, Alison. That was really fascinating and interesting. And I hope everyone was able to take away something valuable for themselves, for their practice. And thank you all participants. I look forward to the next session.